Okay.
Yeah.
Just setting things up. I just popped in now.
think we're still waiting on Lisa. Do you have Lisa's number? It's only like 103.
Are you expecting her?
She didn't stay, so I kind of assumed.
The rest of it?
I just got off the phone with Lisa. She said she's working on getting one.
Thank you. There we go. Okay. So we're ready.
Is it adjustable? I can kind of crouch.
Okay.
There we go. All right. Well, I'm Thomas Nasalaris. I'm Maggie's thesis advisor, and I want to start by thanking everyone for attending physically and virtually. Everyone's streaming online. Just a few procedural notes. So Maggie's going to present her thesis in just a few minutes. Afterwards, the physical and virtual audience members who are not on the committee will exit, and then we're going to switch to a private WebEx link for the committee. And at that point, the thesis offense will begin. That usually takes a while, so yeah, you'll have some time to go before we release her back to you. Okay, well, it's really an honor to present Maggie at her own thesis defense. She came to us in 2015, and I remember her interview. I remember her interview for graduate school. She quite well. One of the things I remember is her describing what was inspiring her to leave a career, a successful career, to completely change course and become a neuroscientist. That's a really risky thing to do. Science as a risky and just kind of an inherently uncertain problem. So I think that took a lot of courage. I've since learned that that is an attribute that's just kind of baked into her personality. She's brought it to bear just multiple times as we've gone down a lot of very twisty and uncertain paths, examining all kinds of interesting things. But 5 years later, she is in fact a neuroscientist and a remarkably capable and insightful neuroscientist who has a really extremely bright future. I and all of her committee members really feel strongly about that. She's developed a really impressive technical arsenal, and she's applied it to actually a lot of really interesting projects that she's presented at conferences all over the world. And most recently, she was invited to contribute a talk to a global audience at the Neuroknowledge Academy, which was extremely well received. And so she's going to talk about just one of those projects now. It's part of a paper that's currently in review. We expect to publish soon. After her thesis, I'm really happy that she's agreed to let us keep her for just a little bit longer while she writes up some of the other work she's done and decides which lucky lab is the next to benefit from her talents. Okay.
Okay, thank you. That was a lovely introduction. So thank you everyone for coming today and virtually. For those of you on WebEx, I'm going to have everyone muted to begin with. I'll keep an eye on the chat to see if anyone has questions during. I'll do my best. Okay, so let's get started. So today I'm going to be talking to you about investigating the source and structure of unexplained variance in natural scenes fMRI data. We'll just go over a quick outline. There's going to be an overview in which I'll go over some definitions, some background, and I'll tell you about the problem that we're looking at today. In the methods, I'll tell you about our data and the models that we're using. We'll go over results, and I'll do that by asking a series of questions and then providing results as answers. I'll tell you about some preliminary findings that we've recently worked on, and then we'll wrap up with a conclusion. So the title of the talk is Investigating Unexplained Variance. So what do we mean by unexplained variance? Let's say this squiggly line here represents a signal that we've measured over time, any sort of signal. And we want to know how it behaves in the future. So we're going to look at the data that we have. We come up with a model that predicts how this signal behaves, and we make a prediction. And we compare that prediction to the data. Now you see this prediction is perfect. It explains all the variance, all the different changes in the measurements that we recorded. But of course, that very rarely, if never, happens in real life. So let's look at another prediction. So here we see that this prediction, it gets the general shape right, and there's areas in which it matches up perfectly with the data that we have, but there are some differences. It declines too soon, it goes too high over here. And the differences between our prediction and our data is unexplained variance. That's just what our model isn't picking up on. And so that's what we're gonna investigate today. And we're looking at unexplained variance in fMRI data. So what exactly is fMRI? Well, as Thomas mentioned, I'm a neuroscientist, so I want to record activity from the brain. And in humans, we generally need to do that in a non-invasive way. So we use an MRI machine. And very simply, a subject lays in the scanner, completes a task, and we record changes in the magnetic field that's in the scanner. And we use that signal to infer brain function. So fMRI is an indirect measure. We're not directly recording neuronal activity, but over the past 30 years, it's been widely used in neuroscience with great success. And today, we're going to focus on the visual system, which is at the very back of your brain. So in visual neuroscience, a critical measure of understanding is the ability to predict how the brain will respond to complex stimuli. And here we see an example of what we mean when we say a natural scene. And the way that we do this is we build encoding models. So an encoding model will take the stimulus that we've presented to the subject. It'll apply some mathematical operation to the features of that stimulus, and it generates a prediction of how the brain will respond. And currently, the most accurate encoding models for predicting responses to stimuli in visual cortical areas are based upon deep convolutional neural networks. So here we see a schematic of a DCNN, commonly known as AlexNet. It's one of the most widely used DCNNs in visual neuroscience. And actually, DCNNs were proposed decades ago by computer scientists, and they were initially modeled off of how we thought at the time our visual system worked. So you can see here we have input data, and then we have all these layers, and each layer performs a different mathematical operation on the stimulus, and then sends it to the next layer. And that was how we at the time thought that our brain works, our visual system works. You have different areas in your visual system, each one looks for a different part of the stimulus, and it builds up this representation of the world as you go up the visual hierarchy. And these models are able to predict fMRI bold activation in voxels all across the visual hierarchy. So we feed our stimuli through this AlexNet, we extract out the features that it picks up on, and we use that to predict brain activity to the same pictures that we showed our subjects. And here we have an example of one of the first studies to actually use this method. So these are two different subjects. And what we're looking at here are cortical flat maps. So it's essentially if I lopped off the back of your brain and squished it down. It's just flattened out. And then we project our data onto that. And what we're looking at is prediction accuracy. So prediction accuracy is how well our prediction matches up with our data. And the darker areas correspond to a low prediction accuracy, and it goes all the way up to 1, which is the highest, and those will be the lighter areas. And we see that there are some parts where we have a medium, fairly good prediction accuracy, but there's a lot of areas here where the model completely fails. And in particular, you'll notice in these middle areas, which is the foveal representation, when you look at something, what's right in the direct center of your view. So these models are impressive. They can predict bold across the visual hierarchy, and they use one underlying model of computation. But these models, and to our knowledge, all encoding models fail to predict brain activity to natural scenes in most voxels. And so that means they can't account for a large portion of the variance in the signal that we are recording. And that's what we're interested in looking at. So what could the source of this variance be? Kind of an initial assumption is that it's just noise. It is an uninteresting signal that we don't care about. We know that biological processes are inherently noisy. fMRI is not a direct measure of neural activity, like I mentioned earlier. And so perhaps we are at the fundamental limit or the noise ceiling of what we can explain in the visual cortex with current fMRI protocols. But that would be fairly uninteresting. And there's a lot of activity that we're not explaining. So it's unlikely that we can just disregard this and think of it as noise. Another kind of obvious option is that we're not including the right features of the stimulus in our models. So DCNNs learn a wide variety of stimuli-based features, so ranging from simple Gabor wave-like ones, like these. This is looks like what we call a Gabor wavelet, and they essentially look for edges in a picture, which is similar to what V1, the first section in your visual system, does. And then they go all the way up to these very complex and strange object-like ones. There are thousands of parameters in these models. We don't fully understand how or why they're learned. So maybe while the features learned by DCNN are great for object recognition in artificial systems, they aren't the best representation of what happens in visual cortex. And certainly that's probably true, at least a little bit, for some later areas that use these complex representations that we haven't quite figured out in the brain yet. But we do know how the early visual systems respond. So it wouldn't explain the inability to predict the early areas, and it wouldn't explain why these models fail, particularly in the phobia. So there could be a third option. Is this unexplained variance not directly related to the features of the stimulus, but also not an uninteresting noise signal? And we can think about this by thinking about what happens when we acquire data with no visual input. And that is something that we do in resting state. So a resting state fMRI scan is when you lay in the scanner and you don't complete a task. You just lay there, eyes closed or eyes open, and you let your mind naturally wander. And via resting state scans, we have found multiple networks in the brain that are functionally connected. And here we see two of them that correspond to visual areas, visual related networks. We have early visual, primary visual, and extrastriate visual. And the activity measured is spontaneous and unrelated to the features of the visual stimulus because there is no picture that you're looking at. Can this tell us anything about unexplained variance present in our natural scene experiments? Well, we know that this signal is highly reproducible across resting state studies. And it's not just a noise signal. Interestingly, when resting state first came out, it was actually thought to be noise. And then in 1995, Biswell and colleagues showed that it was functionally relevant. So it's spontaneous activity, and it does have functional significance. It's also consistent with underlying anatomical structure. Resting state activity patterns have been shown to follow topographic principles for the area being studied. So in the visual system, the world that you see in front of you is mapped onto your visual system in an organized manner. So if one neuron or an fMRI voxel responds to this part of visual space, the one next to it will respond to the part of visual space next to it. And in resting state connectivity, we see that seeds in visual areas have greater connectivity with areas that share the same visual properties, such as visual field location. It's also recently been shown that resting state patterns have a higher correlation with functional connectivity patterns derived from natural scenes tasks as compared to a task showing synthetic stimuli such as faces or objects out of context. So you can come look at these connectivity patterns in resting state and task. And it looks like with resting state, it's more similar to what happens when you are seeing something that's ecologically valid, that you would see in your daily life, than something that you would like a random object out of context. So the spontaneous activity in resting state is not noise, it's highly structured, and it correlates with activity elicited from ecologically valid stimuli. And this is a good indication that the variance we can't explain in natural scenes task is not just noise, but contains meaningful information. And we think our hypothesis is that this variance is stemming from endogenous processes within your brain. And we know that there are endogenous processes that affect the visual system. For instance, mental imagery is completely intrinsic to your brain. You don't have a stimulus. If I ask you to imagine a dog and you picture a dog in your brain is generating that. And it's still using parts of your visual system to do that without a stimulus. We also know that memory and attention can feed back into the visual system and affect how you perceive the information coming into the visual hierarchy. Again, indicating that our missing variance is meaningful. The structure of purely fee-forward DCNs like AlexNet don't have a mechanism to account for this variance stemming from internal sources. And so today we seek to investigate the mystery of missing variants. And we're going to do that by looking at a data set that is publicly available. It was acquired at University of California, Berkeley on a 4T scanner. Subjects were shown stimuli like this deer here. They're data from 2 subjects, and the coverage is right in the back of your brain, 18 slices right in the back of your brain. We're also going to look at another data set. So this data set was acquired with subject 1 from the previous study on a 7T scanner at the University of Minnesota. And you can see we have these bars and these wedges, and they're filled with objects and faces and different things. And the bars sweep across the visual field in all different directions, and the wedges go around and rotate in both directions. And what we use this for is to estimate the receptive field location, which is the topography that I was talking about earlier. And this is going to be our ground truth receptive field information. So this is going to be what we compare our other models to. And speaking of models, since I told you a little bit about the stimulus to voxel model, it's not explaining variance, so we need to think of something else to use. And what we're going to use is called voxel to voxel modeling. So whereas the stimulus can only capture variance associated with visual characteristics of the stimuli, for instance, where diagonal edges appear, voxel to voxel models rely upon the correlation of activity between voxels, and therefore can exploit endogenous activity. And in this equation here, W is a weight matrix of for each source voxel, and R source is the array of source voxel activations, and R hat target is the prediction of our target voxel. We use ridge regression to determine the weights assigned to each source voxel. And so essentially what we're doing is we're looking at one area in our visual system, taking all the activity from it, weighting it, adding it up, and coming up with a prediction for another target voxel. And we're going to do this in 6 areas, V1 through V4, V3AB, and LO. And we're going to build models for each and every voxel based on every source area. So if we have V1 predicting V3, we're going to consider that a feed-forward model because it moves forward in the hierarchy. If we have V4 predicting V2, we consider that a feedback model because we're going back in the hierarchy. And then we can also have the instance where all of the voxels in LO are used to predict another target voxel in LO, and that would be a lateral model. And we're going to compare this to our stimulus to voxel model. So our stimulus to voxel model also uses AlexNet. And for natural scenes data, we apply this, we feed the stimuli through AlexNet, and we extract out the resulting feature maps. Candidate receptive fields, which is this grid right here, to determine which part of the feature map activations are regressed against voxel activity for the same stimuli. So you can see here we have this receptive field center as a candidate, we have these sizes are a candidate, and we combine them, we look at the information just in that area of the feature maps from the stimulus, and we use that to predict brain activity. And we do that for every combination of receptive field center and size. And the combination that has the highest cross-validated prediction accuracy is considered the receptive field location for that voxel. And here is another cortical flat map. And you can see that our model can predict many voxels throughout the visual hierarchy with high accuracy, better than the model that I showed you earlier, but there's still a lot missing, especially in these foveal areas and some of the later areas, it is a lower prediction accuracy. Okay, so let's dive into how we are investigating this problem. First, we're going to think about the source of unexplained variance. As I mentioned earlier, our hypothesis is that the source is endogenous. It's coming from within your brain. And we're going to ask 2 questions. One, is the source of unexplained variance unique to each voxel or common source shared across voxels? And 2, is the source of unexplained variance unique to each brain or shared across subjects? So in the first question, what we're looking for is the activity shared across the cortex? If it was an endogenous source, you would expect it to be affecting all of the visual hierarchy. If it was something like noise or stimulus feature that we're missing, that would be individual to each voxel. And is the source of unexplained variance unique to each brain? So if it is endogenous, it should be unique to your brain, because I shouldn't be able to have the same endogenous activity as someone else. Okay, so let's look at the first question. Is the source of unexplained variance unique to each voxel or a common source shared across voxels? So again, we're going to use our voxel to voxel model here. And we're going to have a grid comparing our voxel to voxel model to our stimulus to voxel model. So we have the source areas are along the rows and the target areas are along the columns. So for instance, this plot right here is V1 activity predicting V2 voxels. And we're going to have voxel to voxel prediction accuracy on the x-axis and stimulus to voxel prediction accuracy on the y-axis. And you can see for all of our source target pairings, our voxel to voxel model out predicts our stimulus to voxel model. It's explaining a lot more variance than our stimulus to voxel model is. And you can see this also in our cortical flat maps, that across the visual hierarchy for each source area, there's high prediction accuracy and very little, if any, voxels are missing or not being able to be predicted. So from this, we can infer that, in fact, the activity that we are explaining with voxel to voxel models is shared across the visual hierarchy. So our next question, is the source of unexplained variance unique to each brain or shared across subjects? So what we're going to do here is we're going to take all the activity in subject 1's brain, and we're going to use it to predict a target voxel in subject 2's brain. And vice versa. And we're going to do this with all the same source target pairings that we did before. And we're going to see the same exact grid that we saw previously, only this time there is no advantage. Voxel to voxel models built on someone else's brain can only predict as well as the stimulus. So my brain can only predict your brain as well as the same thing that we're looking at. And so is the source of unexplained variance unique to each brain or shared across subjects? It is in fact unique. And both of these lines of evidence point to it being an endogenous source of activity. But that doesn't mean it's not noise. So noise can also be endogenous. Which brings us to our next set of questions. Is the source of unexplained variance physiological or machine noise? Is variance explained by voxel to voxel models retinotopically mapped? So physiological or machine noise, when you're in the scanner and you're breathing and your heart rate can actually interfere with the signal that we're reporting from. So we're going to look and see if that's actually what's causing our voxel to voxel models to outpredict our stimulus to voxel models. And is variance explained by voxel to voxel models retinotopically mapped? We're going to look to see if there's structure in this variance. Is it meaningful? Can we extract information out of it? So we'll look at our first question, and we're going to go back to our voxel to voxel models, but we're going to come up with 2 more ROIs. So the first ROI up here is in white matter. And so white matter would be affected by the same physiological processes as the gray matter, but it shouldn't contain neural activity. It shouldn't be able to predict neural activity. And then we're also going to look at air. So we're going to record activity in the space around your head while you're in the scanner, which also, of course, should not affect your neural activity. We're going to take the predictions that these 2 ROIs make, and we're going to subtract them out of our original data. So we're going to take any variance that is explained by one of these ROIs, we're going to take that out and see if we're still doing as well as we were before. And so I just want to remind you, this is what our original model with all the activity looks like. And so what we want to see here is we want to see very little change when we subtract out white matter or air voxels. And so we'll look at white matter first, and we see there is in fact very little change. When we take out any variance explained by white matter, we're still able to explain a lot more variance than our stimulus to voxel model. And it's the same with air. So taking out any scanner or machine related noise in the signal, doesn't affect our ability to predict with voxel-to-voxel models. So is the source of unexplained variance physiological or machine noise? It is in fact not. Is variance explained by voxel-to-voxel models retinotopically mapped? So how are we going to figure that out if voxel-to-voxel models aren't using retinotopy? We're going to combine information from our two models. So here's our voxel-to-voxel model. Here's our stimulus-to-voxel model. In the stimulus-to-voxel model, we're going to use specifically the grid, the candidate grid, and we're going to turn that into a 2D histogram. So this is basically visual space broken up into all these candidate receptive field locations. We're going to look at our voxel-to-voxel models, and we're going to pull the weights from the source voxel. We're going to pull the weight, and then we're going to put it in the same location that the stimulus-to-voxel model predicted that voxel likes and cares about. And we're going to do that over and over and over again for every single source voxel in a target voxel's model. And then we're just going to add up all the bins, and we're going to select the one that has the highest sum of weights. And that's going to be what we consider our voxel-to-voxel receptive field. And we're going to compare this to both our stimulus-to-voxel receptive field and our ground truth receptive field. So here we see V4 source activity predicting V1 target voxel. So all of the activity in V4 predicts one voxel in V1. This target voxel had a 0.02 prediction accuracy in the stim-to-vox model. The stim-to-vox model was not able to predict it. But our voxel-to-voxel model, it had 0.66, so it had a much higher prediction accuracy in our voxel-to-voxel model. Each one of these squares represents visual space, so it represents the amount of visual space that the picture you're looking at took up. We have in teal is our ground truth receptive field. That's the one we estimated from our retintopic mapping experiment. In gray, we have the stimulus-to-voxel receptive field. That's the one we estimated using our fourth model. On the top, we have the 30 largest and 30 smallest weights, and they are coded by size and color. So a big red circle means that voxel was given a high weight in the model, and a small dark circle means that voxel was given a big negative weight in that model. And we can see that in the positive weights, the voxels tend to surround and cluster around the ground truth receptive field. Whereas in the negative weights, they tend to be on the periphery. And then here we have all our source weights plotted, and we take all those source weights, again, we add up the bin that they're in, and it looks something like this. And then we select the one that has the highest sum, and that is the one that's outlined in yellow here. And you can see that, in fact, our voxel-to-voxel receptive field is closer to the ground truth than our stimulus-to-voxel receptive field. And we see this with all sorts of combinations. So here we have V2 predicting LO. And in fact, our stimulus-to-voxel model predicted that the receptive field was off to the side. But in fact, our voxel-to-voxel model is able to pick up that it's closer to the ground truth. We see, again, V1 and V3, very similar. The receptive field is off to the side, but we are much, much closer to it with voxel-to-voxel, and LO to LO. So it looks like this variance is retinotopically mapped. But these are just a few examples. I could have just picked the best ones. So we're going to look at this and see our voxel-to-voxel receptive field is really closer than stim-to-vox. Actually, I'll go back. What we're going to do is we're going to measure the distances. We're going to measure the distance between the ground truth and the stimulus-to-voxel receptive field for every target voxel. And we're going to measure the distance between the ground truth and the voxel-to-voxel. And we're going to plot those distances on the x-axis as a histogram. So it's a distance between our predicted receptive field and our ground truth receptive field. We're going to compare those distances for our two models. So here we see all the voxels as they were predicted by V1 source activity in orange, and compared to as they were predicted by the stim-to-vox. And this is just for voxels that were not able to be predicted by our stim-to-vox model, but were with our vox-to-vox model. So you can see none of these voxels reach threshold in stim-to-vox, and they are tend to be pretty far away from the ground truth. But in our voxel-to-voxel model, most of them are within 5 degrees of the ground truth receptive field. We see this across all source areas. And so again, it looks like this is retinotopically mapped. However, there's still stimulus information in the signal, so perhaps the voxel-to-voxel models are just amplifying that or picking up on that. So how can we look at that? What we're going to do is similar to the white matter and the air voxels, we're going to take out that information. We're going to take the predictions from our stimulus-to-voxel model, we're going to subtract it from our data and rerun all our voxel-to-voxel models. We'll recalculate our receptive fields and our distances and see what happens. So these are going to look very similar. Now we're looking at voxels all across the visual hierarchy. And even after removing stimulus information, there are still more voxels that are closer to the ground truth receptive field than they are in our stimulus to voxel model. And again, this is across the visual hierarchy. So is the variance explained by voxel to voxel models on the top of the map? Yes, it is. So what have we learned so far? Simple linear voxel to voxel models can account for fMRI bold variants that highly optimize nonlinear stimulus to voxel models are blind to. The signal is shared throughout the visual hierarchy, but not between brains, indicating an endogenous source. The signal is retinotopically mapped and cannot be accounted for by nuisance sources alone. And the signal is highly structured, meaningful, and perhaps exploitable by a new generation of models that account for endogenous activity. And so this is pretty exciting. So we have all this variance that we can explain, but it's just in this fMRI signal. And fMRI in each voxel are thousands of neurons. We're summing over that activity. So perhaps this is actually just something that stems from the way we were measuring it and not something that is actually really in your brain and related to brain activity. So we can ask what happens with neurons. And as it turns out, last year, a data set was released with mice and 2-photon calcium imaging. So these mice run on a trackball and look at these 3 monitors here with natural scene images on them. All the natural scenes are related to what a mouse would see in its normal life. So this is a bird. And these mice are bred such that when they release calcium, there is a fluorescent signal. So there's a green light that comes out, and calcium is released when your neurons fire. So we can tell where and when neurons are firing while they're looking at this stimulus. And so we're going to do the same exact thing that we did in fMRI. First, we're going to look at stimulus to neuron. So we're going to take our exact PORF model and apply it to our neuron data to see if it works here. We're going to apply it in the same way we did with the DCNN, and we're also going to use a set of Gabor wavelets as features to see if it works with that type of feature as well. And so what we're looking at here is the comparison of those 2 models, DCNN PORF on the bottom and Gabor PORF on the top. They both do a pretty good job of predicting activity in calcium imaging, and it looks like the DCNN does a little bit better. We can also look at the receptive field locations that they predict. So these are a representation of the 3 monitors that they looked at. These dark squares are the receptive fields that were estimated by the original paper, and they did a coarse-to-fine procedure where with Gabor wavelets in order to estimate these receptive fields. And because it's computationally intensive, they had to kind of restrict it to a smaller area. Our model is very flexible, and so we looked a little bit further out. You can see that a lot of neurons do, in fact, in both models, fall under the same receptive field location as the original paper, but we also have a large population that fall outside of it. And so perhaps because our model is more flexible, we're able to more accurately predict for some of these neurons. And from here on out, when I compare anything, we'll be using the DCNM model. So now we can build our neuron-to-neuron models. And again, our neuron-to-neuron models are just like our voxel-to-voxel models. This is all in mouse V1. We're using all the neurons in V1 to predict one other neuron in V1. And we can look the same way we did before with our neuron-to-neuron model prediction accuracy on the bottom and our stimulus-to-neuron model prediction accuracy on the y-axis. If we look at all neurons, it has very, very, very similar shape to our lateral models in our voxel-to-voxel method. So our neuron-to-neuron method is able to increase the amount of variance that we are able to explain over the stimulus-to-neuron method. Some of the mice actually also had a second fluorescent reporter that allowed them to identify interneurons or inhibitory neurons. So we can separate that population and see that most of the neurons were excitatory. So the excitatory plot looks a lot like the all plot. But the inhibitory plot, we could see that it appears as if inhibitory neurons might be see even more of an advantage in these neuron-to-neuron models. And then, okay, so we did cross-subject in our voxel-to-voxel. So now we're going to look at cross-mouse in our neuron-to-neuron. I'm going to use all the activity in one mouse V1 to predict a different mouse's neurons. And again, we see very similar pattern to what we see in our fMRI data, in that one mouse can only predict neuronal activity in another mouse as well as the stimulus that they're looking at. And if we break that out, we see that that's true for both excitatory and inhibitory neurons. So this is a pretty exciting finding that we're looking at two very different levels of measurement in two different species, and we're coming up with very similar patterns, indicating that, in fact, ongoing and spontaneous activity is part of the signal that we're reporting in our task. And I mentioned earlier, spontaneous activity in fMRI is recorded with resting state. Our original data set did not have resting state, but over the past 2 years, our lab, in collaboration with another lab, has collected more natural scenes data sets with more subjects. And we also collected resting state. So I'm going to take that data, I'm going to build the voxel-to-voxel models in the natural scene experiment, exact same way that I did with our original data. But now I'm going to use the resulting models to predict resting state activity and see what happens. I'm going to compare how well it can predict natural scenes and how well it can predict resting state. And so we see here with resting state prediction accuracy on the bottom and natural scene prediction accuracy on the top, on the y-axis, that we can predict resting state just as well as natural scenes. Even though these models are built when you're looking at pictures and built on visual stimulation, we can still predict resting state with high accuracy, just as high accuracy as we can the natural scenes, indicating that the processes, the activity that are going on in both the task and resting state are linked. And the mouse experiment also recorded spontaneous activity, so they just let the mouse run on the ball and just recorded the neurons firing during that. And so we can do the same thing. We can compare the predictions of our natural scene data to the predictions during spontaneous activity. We see a very similar pattern. Most of the neurons are well predicted, or predicted as same in natural scenes as resting state. There is this chunk out here that is better predicted during the natural scenes task. And if we split that into excitatory and inhibitory, we can see that it looks like most of those neurons are excitatory, which is interesting and a possible direction to explore in the future. Although I will say, again, this experiment was not targeting inhibitory neurons, and there's a lot less of them, but it's just a possible avenue that we can explore in terms of inhibitory versus excitatory and spontaneous activity. So let's wrap up with some conclusions. You often hear that the brain is noisy, and I alluded to that even in the beginning of my talk. But up until recently, if you asked a random neuroscientist what is noise in brain activity, they would probably say anything not directly related to the stimulus. I think we've shown here today that that's simply not the case, and perhaps we need to be more careful about how we think about noise and signal in brain activity. So rather than saying it's noisy, we can say that the brain is a complex dynamic system. And in network neuroscience is a field that's emerging as a promising approach to studying the brain. In the past year alone, there's been a lot of data across species and methods showing that ongoing activity is not only the predominant portion of activity that we report from the brain, but also functionally relevant. Which brings me to my next conclusion is that ongoing activity is a feature, not a bug. Ongoing activity uses up the large majority of the brain's metabolic resources. It would be highly inefficient to expend that amount of energy on processes that are meaningless. So rather than thinking of the brain as a reactionary sensory processing machine, we need to really think of it as integrating incoming sensory information into an already active and rich perceptual experience of the world. And that shapes how we process sensory information rather than the other way around. This allows neural processing to be highly flexible while remaining computationally efficient. Finally, ongoing activity is present and measurable and modelable at both micro and macro scales. So we're able to isolate and model ongoing activity in both fMRI and 2-photon data with very, very similar results. And this has some important implications. First, it should make us consider how we design experiments and interpret our data. So for instance, often in visual neuroscience, we consider differences in how the brain responds to the same stimulus over repeated presentations as nuisance, something that we should be ignored. But perhaps this, we can actually exploit this trial-to-trial variability to provide insights into the role of ongoing activity in the brain. And we should be careful to interpret data from experiments with synthetic stimuli, as it may not be integrated into ongoing activity in the same manner as ecologically valid stimuli. We can also think of this as having some clinical implications. So for instance, abnormalities in the visual experience are present in disorders that produce hallucinations, such as schizophrenia and PTSD. And developmental disorders like autism and ADHD often present with abnormal, sometimes painful reactions of bright lights and other aspects of the visual world. So instead of investigating these abnormalities and how they're processed, how this incoming sensory information is processed, we may need to investigate the differences in ongoing activity and how that sensory information is integrated into the brain's dynamic system. So with that, I would like to say thank you to my advisor, Dr. Thomas Aslaris. He has taught me a lot over the years and has had a lot of faith in me, and I truly appreciate that. My committee for not only providing valuable feedback, but building my confidence in myself as a scientist. Our funding sources from the NIH and the NSF. Our lab, particularly Giuseppe for having an infinite amount of patience with me. And Jesse for just being there for me the entire time and becoming one of my closest friends. Our collaborator, Kendrick, is responsible for acquiring the NSD data and processing that. And so he has also been a big help. I'd like to thank my parents for supporting me through this very crazy journey. And my family and friends for also being there for me. And that's it. I don't know if anyone in the chat has questions, but if not,
Yeah, if anyone in the audience wants to ask questions, feel free. Otherwise, we will. Oh, here we go.
of course you can, Emily. Thank you. Mm-hmm. Mm-hmm. Mhm. Yeah, no, I get what you're saying. So Emily's asking, it seems counterintuitive that the stimulus to voxel model is unable to model voxels in the fovea because that's where we get the most activity. But actually, there's been a lot of research that shows that feedback like attention and working memory predominantly affect the foveal representation, which actually, if you think about it, kind of makes sense. So if we're thinking about like a predictive coding framework, you would want to predict the area that you're seeing the most to use the most efficient resources. So, and also there is an MRI, but like the vasculature and the SNR sometimes in the fovea can be a little bit less. So that's probably affecting it too. But I think it's probably the ongoing activity is having a big effect there. Okay, I don't think anyone on the chat has questions.
Okay.
Okay.
Well, thank Maggie one more time for beautiful presentation. Thank you. The private.
Yeah, if the committee members can just, I'm going to end this WebEx and we can go to the private WebEx, the second link that I sent out. I'll start that now.
Okay, great. Yeah, so we'll, Maggie, wow. So, yeah, so this is the part where I'll exit.